Hello, my name is Bijan Barenji, and I'm here to help you to find the information you need for your project in Phys. Once you know what gene you are working with, come to NCBI, click on the drop-down box menu, and go to Gene. Type in the gene that you are working with. Next, you're going to want to click on the gene that is associated with Homo sapiens. If you do not see it on the first couple of the pages, go ahead and go to the right hand side, and Homo sapiens should be somewhere here. If not, click on the More tab. Next, this is where you're going to find all the information you need for part 1. So, let's look at the template. We know the name of my gene, which is HbA1. The gene function, it codes for hemoglobin, that is the protein. Next, we're going to look for chromosomal location, nucleotide range, number of exons, and SNPs. That's all on this page. Chromosomal location is right here. Exon count is right below it. Then we're going to come down to this map and hover right above HbA1, my gene. You'll see the location. The nucleotide range is right here. Next, we're going to find the SNP count. Scroll down a little bit. It is right here. This will give us the total number of SNPs. Once this loads up, we're going to put everything in those boxes. Well, I already have, but you'll put your SNP here, exon and nucleotide range, and your chromosomal location. For this box right here, it's a completely different website, and I will show y'all. This is where you find out. 365. That's my results. Here on out, I'm going to be working on Snippepedia. I'm going to go type in HbA1 in the upper right hand corner. These websites provide information about the gene. You can also find some of your extra information here possibly. 23andMe, Google, or Wikipedia would be good. These are snippets that may or may not cause a change in the normal function of the protein. We are only looking for snippets that change the normal function of the protein. For example, this right here. This snippet, a new variant with increased oxygen affinity. So I'm going to want to put this snippet in my template, which I already have, right here, and I put what it did. Now this is the important part of putting the genotype, CC altered hemoglobin. This is where I got that from. Okay, but there's 3. You need to put however many there are that changes the function. Okay. Next. You get the example right here. The next one, but I didn't put the genotype here. The genotype does need to be on each one, and you can always find them in the same spot. That's it for part 1. Part 2 is all about the protein, which is a product of the gene. That is something that everyone needs to understand, that a protein is a product of a gene. In the past couple of semesters, there's been confusion on what goes in this box, in this section, and it is only information about the protein. I'll show you where to find all that information. So the first thing is the name. So with the name, we can go here, hemoglobin alpha 1, or I can show you, this is another part, official full name, or I'll show you where we'll get the majority of the information for part 2, and it also shows you a separate name. A completely different name, but it is still acceptable right here. So we're going to want to know the length, which is 142. Make sure you put amino acids at the end of that. And the mass, which is at 15,500 258. Daltons, or Da. That's where you find this. Okay. Next, subcellular location. Just click right here, subcellular location. This is the information right here. Next is post-translational modification, which is right here. Amino acid modification. This is where you put this. You put how many of each feature key there are. There's 10 modified residues. And I believe, okay, there's 4 of these glycolizations. And put what kind. If you want to get really specific, put the position. Next, actually, I believe that is all for this website. Let me make sure. Cytoplasm. Yep. Okay, next I'm going to show you where to find the domain. For domain, we can go back to Snippepedia and just go ahead and open up Wiki. And this is where I found my domain on most of my genes that I've done research on. This is where I found it, right here. It's safe to get the information from here. If you don't feel comfortable, look up a different website and see if it comes back as the same thing. Now, we're going to go to a new website to find tissue expressions. It is called proteinatlas.org. Type in your protein here. Click on the correct protein. This shows you where the protein is transcribed in the body, and if it's a high amount, medium amount, or low amount. And it gets pretty specific because it even talks about what cells they are transcribed in the most, which is pretty cool. So you can get pretty specific on this if you're wanting to, or if they're wanting you to get pretty specific. Next, it's going to be extra. You can do this easily. Go to YouTube, go to the library, use some of their database research engines, and try to find something interesting, something that catches the eye. Protein function can be found very easily. Go back to this one. Click on function. Bam. Involved in oxygen transport from the lungs to various peripheral tissues. Right there. This can also, on the Human Atlas, can also give you description sometimes, or even right here. So check these out. And the description on the Protein Atlas may be the same as the one on NCBI, but that's okay. Sometimes they're different. But this is where you'll find information. Or sometimes you can even find little articles that will be like here on the side that may help also. And I believe that is all. Dr. Wilk will be talking about part 4 with y'all in class more extensively. I just told her I would cover part 1 through 3 because I know the past couple of semesters students have struggled finding the information. I hope this video helps, and good luck with your project.